Alrighty, I'm just checking off the people who are on Zoom. We're we few but mighty in person today. We do have a few people on Zoom, but other folks are wherever they need to be, and some will show up and some won't, and that's okay. Alrighty. Okay, so we talked about the different disorders of the sensory system on last Wednesday. And today what we're going to focus on is the sensory system itself. And so I know some of you all took sensation and perception with Dr. Ariel. So to be perfectly honest, you all know more about sensation and perception than I do because I never took a full S&P course. But I do know the stuff relevant to neuropsychology. So I'm going to use a bunch of visual aids to show those off today, including some stuff from my intro PowerPoint and some stuff from the book chapter PowerPoint. So we are good there. All righty. So the sensory systems, there's obviously more than one, and they are fairly complex. So each sense has very slightly different receptors, and the ones for vision are photoreceptors that take in light. The ones for hearing are actually kind of touch receptors, because what happens in hearing, and we'll go through this in a lot more detail in a few slides, is it's actually sensing the vibrations that the rest of your ear translates the sound in. Touch is obviously a big one. And the smell and taste are chemoreactors, so they are both reacting to various chemicals that are there. And so, again, this is going to focus a little more on the actual process of sensation, but what happens in neuropsychology is that somehow the communication between your sensing organ and the brain itself is interrupted, or the part of the brain that usually makes sense of that sensory part is damaged in some way. And so, I'm not going to get into too much detail here, but this is your eye. And what happens is that the light comes in, hits your lens, the image is actually reversed, and it hits the back of your eye, the fovea. So on the fovea, you have all your rods and cones. You can recall back to Intrapsych or S&P if you took that. Cones detect color, rods detect sort of light and dim and nuance there. And the whole fovea is covered with those, with the exception of the blind spot. Called the blind spot because there are no receptors. And what happens at the blind spot is your optic nerve leaves the eye. You need a place for the optic nerve to leave the eye. So that's your blind spot. Luckily, our vision replicates itself, so both eyes see sort of this little almost like windshield, and then they cross. So, you know, one blind spot can't see, the other eye can fill in, then we get one continuous picture. In the vast majority of situations, right? Obviously, there are other things that can happen. Yeah, I just mentioned all those. All right, and then this is getting a little more technical about what happens in the fovea. You have the light come in the back of the retina. And what happens is that, again, it goes through these rods and cones. The rods and cones, several of those transmit information to one bipolar cell. Then the bipolar cells transmit information to the ganglion cells. The axon of the ganglion cells, or axons of the ganglion cells, are what make up the optic nerve. So this is what goes back into your brain to communicate with the visual cortex. So again, it's a bunch of axons of neurons, essentially. And this is getting at what I was trying to explain about how our eyes overlap. In terms of what we see. And so each eye sees both the right and the left side. It's flipped for both eyes because of the way the lens takes in the light. It goes back to the visual cortex and reflipped. Here you see the optic chiasma, which is where they cross, so that all of the information on the right side is taken to the left side of your brain, and all the visual information on the left side is taken to the right side of your brain. Again, that's your optic nerve. And then you see some of the parts they were talking about, so the lateral ventricular nucleus and the primary visual cortex is back here. And so we mentioned this in passing before, I think. The corpus callosum is the main point where information crosses over from the two hemispheres of the brain, but they might sever that in order to prevent seizures from spreading, things along those lines. It still transmit information, although not as efficiently, because it's not really what it's made for, over the optic chiasma if that happens. So light itself is just a very small, or at least visible light. It's just a very small portion of all of these existing wavelengths that include things from gamma rays up to AM radio. All right, so again, just kind of touching on each of the senses here. So talking about hearing, what we are going to be talking about is the physics of sound. So the frequency is the number of cycles completed by a wave of sound in a given amount of time. So this wave has relatively low frequency, and this wave has high frequencies, more cycles being completed in the same amount of time. Amplitude is the physical strength of the wave. So we could have high versus low amplitude. These are going to be related to loudness, but also to some of the quality of the sound. And just for context, if you've ever heard of decibels, right? So we do pretty well sort of within this range. But if you have hearing loss, or sorry, if you have prolonged exposure above 90 decibels, you can actually have hearing loss. And this has happened to a lot of the boomers and the Gen X folks, probably folks in my generation too, from going to concerts and standing directly in front of the speakers. They'll have hearing loss or tinnitus, which is like a ringing in the ears, but you can get it other ways as well. And this is why you'll see the folks who direct planes, right, on the tarmac, or those giant headphones, they're protecting their ears because you could see how intense like a twin engine airplane or a jet plane takeoff is. Rocket launches, when we're launching into space, even more intense. One of the many reasons they keep people back from them. The others, of course, a bunch of gas and flame come out. All right. And so this is the inside of the ear. And so every part of the ear kind of works in concert to help us interpret sound. So the part of the ear we can see basically helps bubble sound into our ear. It goes into the canal. And then you have the oval window. And there's a series of bones right there that hammer the handle of the stirrup. Right here is the tympanic membrane or the eardrum. So what happens is the waves from the sound come down your ear canal. They are interpreted by the eardrum, kind of like hitting an actual drum. Like it vibrates like an actual drum. Those are in turn interpreted by these little bones that hammer the handle and the stirrup to the ground window. The ground window connects to the cochlea, this fun thing that kind of looks like a snail. The cochlea is filled with a liquid, and as those bones transmit the sound waves to that ground window, it makes the liquid kind of sway. And there's all these little fibers in there. And so those sway. And then that is what the auditory nerve actually interprets as sound takes to the brain. The semicircular canals here are the only part that's not really directly related in hearing. These help with balance. And then the eustachian tube kind of goes down and next to the front. This is why if you see a specialist for ears, you're probably going to see an ear, nose, and throat person. And so here you can see that basilar membrane where all those little hairs are. Here's some zoomed in where you can see the eardrum and then those three little bones of the middle ear attaching to the round window, and then that attaching to the cochlea, the auditory nerve, understanding and basically translating that up to the brain. And so here you can see the vibration sensitive hair cells within that basilar membrane. So our body is doing a lot to interpret inside and outside sensations before we even get to the brain, right? And that auditory nerve, just like the optic nerve, that's that pathway that's going to go back to the brain for you. All right, smell, as I mentioned, is a chemosensor or censoring little bits of chemicals. Olfaction is the fancy name for smell. Olfactory bulbs are the parts that get those particles of chemicals that allow us to smell. Pheromones are something we can put off for each other that can be involved in communication. Animals use pheromones in a variety of ways, including mating, but also to send signals. Bees in particular use their pheromones to send complex signals like the good pollen's over there, or hey, we're all moving to this new hive. Taste, as I mentioned, is gustation, also chemical. And so we have these little papillae on the taste buds. And these are the gustatory cells. Those connect again to axons that become that nerve that takes that information back to the brain. So taste buds are those receptors for taste. They're primarily on the upper side of the tongue. We do have some on the lower side of our tongue as well. The skin senses. So we know that the skin does a lot for us, right? And we tend to just think of touch, right? Like touch is the overarching term we use. But our skin senses can actually do a whole host of things. So not just pressure from touch or that I am touching something, but also warmth versus cold, pain, right? Our skin senses help us to withdraw if something is hot or sharp. But we also have these other senses as well. So the kinesthetic senses use things like those semicircular canals in the ears to help with movement, posture, and orientation. They help with our muscles kind of understanding where we are in space, and that's the proprioceptive feedback as well. And then I mentioned the vestibular with balance and acceleration. So again, these aren't senses we include in like the 5 senses, but they're very important for us to know how to walk and not fall over and things along those lines. Alrighty, so now I'm gonna kind of refer back to your SGA, but I just wanted to give you all a nice little overview of some of that stuff. And so we talked about the different types of sensors that we have. One thing that I didn't mention in particular about touch and the sensors for touch and pain is that it's tissue damage that releases a chemical that activates the pain fibers. And the chemical, even though it's throughout your body, it acts in a similar way to the neurotransmitters in your brain. All of these receptors are pretty narrow bands. They respond to only certain things. And this is why if you have experienced, for example, we're using pain as an example again, if you have experienced very severe damage, at least momentarily, you often sort of feel numb, right? Like a very severe burn or something along those lines. It's your body trying to protect you and help you to get help, right? Without the pain overwhelming you. And so these are specific to these receptive fields. They provide a unique view of the world for each sensory system and locate those sensory events in space, right? Because if our eyes are able to communicate to our optic nerves, hey, I see a bear, right? We need to know, where is the bear? How fast do I run? What kind of bear is it? Because some bears you're supposed to make yourself big, and some bears you're supposed to lay down, and other bears you're supposed to try to outrun everyone else in your group because that's all you can do, right? Or is it a bear on TV, right? And so that becomes really important, is where in space these are. And then we have exteroceptive, which are receptors that are responding to external stimuli. And I think that this is typically what we think of when we think of the senses, right? Reacting to the world around us. But the other really important thing that our senses can do is be interoceptive and respond to what's going on inside our own body. And that helps us interpret the meaning of external stimuli. So for example, interoceptive receptors can tell us whether we're hungry, right? But that can also tell us whether we're full. So you're at, I don't know, Captain George's, right? And everything looks really good, but your stomach says, whoa, please don't eat anything else, right? And you know to stop. Optic flow allows us to process visual information while we move. Wouldn't be much help if we couldn't run or walk without interpreting visual stimuli. And then auditory flow is like the Doppler effect, basically. The changes in the intensity of sound because of our changing location or the location of something else relative to us. All right, and the neural relays are receptors that connect to the cortex through a sequence of 3 or 4 intervening neurons. So oftentimes it's not, you know, like the nerve from my foot isn't going to directly connect to my brain, right? That's not practical. So we're going to have other neurons in between, and maybe even other neurons in between within the brain itself once it gets up there. These also allow the sensory systems to interact with each other, which becomes very important, and to produce relevant actions, right? Drawing your hand back if something hurts, complex emotional responses, or even you're an athlete and you're on the field, knowing how to respond when an opponent is coming towards you. So transduction is where that sensory information is turned into things that the brain can interpret, basically action potentials and neurotransmitter messaging. And the presence of a stimulus is encoded by an increase or decrease in how fast neurons are firing. Basically, the discharge rate sounds kind of weird, but that's what it is. And then the amount of increase can also encode intensity. And qualitative changes are encoded by activity in different neurons. We don't just have one neuron responding. What can happen because these different sensory areas interconnect is that we can end up with some crossed wires, intercommunications. Let me make this bigger because I just realized that font is kind of tiny. There we go. So this kind of shows where everything is coming together. So we have eye movements, we have daily rhythms, your circadian rhythm, sleep-wake cycle, when you're hungry, when you're full, things along those lines. And then you have things like this tells you that muscle that controls your pupil size to change in response to light. You have the pineal gland related to circadian rhythms. We have the superior collis, which in addition to the parts in your ear, help you to know where your head is in space. Very important. Accessory optic nucleus, eye movement to compensate for head movement. And then here's the visual cortex, which is the part we think about when we think about vision. All right, but all these other groups are also related to vision in and of itself. And then other parts that would be coming in, so like here, somewhere around here. So that's kind of coming in at the same place, right? And so these things can kind of cross over, communicate, overall interconnectivity. There we go. There's the word I'm looking for. And so what this results in sometimes is something called synesthesia. And so I have a brief film about that I will open over here for us to look at.
Onions were pink, beer is blue, so peas are purple.
So about 150 years ago, people were very interested in synesthesia, and it sort of dropped out of the scientific consciousness for the last century. And the reason is the school of behaviorism took over, where people were interested in treating humans like black boxes, input-output devices. And you weren't allowed to talk about what it's like to be on the inside anymore. So people stopped being interested in consciousness and what it's like to actually perceive the world. And it's only in the last 20 years or so that the question has re-emerged. So synesthesia has had a real renaissance in the last couple of decades, where people want to understand what is the neural basis of perception, and how is it that some people perceive the world differently than other people? What is it like to see differently from the inside?
And I don't know when I necessarily first became aware of it, because it seems like it's always been there, but I know that I was definitely playing with it when I was 5 or 6 years old, because I remember raiding my parents' record cabinet, searching for records that I liked to listen to for the colors.
Synesthesia is an involuntary, unconscious, automatic linking of perceptions. A stimulus in one sense might trigger a perception in another sense.
It's just sort of always been just the way I think. It's just one has always been yellow, you know, certain letters have always been certain colors.
The days of the week are pretty, well, they're terribly consistent. Monday is red. And my mother said when I was 5 years old, no, Monday's blue Monday.
We don't hear with just our ears alone. We don't smell just with our noses. We don't taste just with our tongues.
Hey, wait a minute, Doc. After 20 years behind this camera, you're not going to tell me I don't see with my eyes, are you?
No, in a strict sense, you don't, Hal. Your eyes, like your other sense organs, simply receive impressions from the outside and send them on to the brain. The senses are all different. The signals are all alike.
Yeah, but then how could the brain know which sense is sending the message?
Phil, you've just asked the big question. That brings us up to the brain.
This back portion of your brain is almost entirely devoted to vision, and there's a particular area of the brain that cares in particular about color. The part of your brain that would be sticking out here cares about language and words and letters, and somehow in a synesthetic brain these areas are connecting to these areas so that words and letters will trigger a color experience.
I have grapheme color synesthesia, and so basically I associate most letters and all numbers with some sort of color. One alone by itself is yellow, but 4 is also yellow, but it's a more golden yellow. Some of the differences are very, very subtle, but they're definitely distinct differences in my mind.
Synesthetic perceptions can be unpleasant every once in a while. But then again, so could your sense of smell. If you're able to smell something, you probably have experienced really pleasant odors like fresh baked cookies, your favorite home-cooked meal, your favorite perfume, and you've also experienced very unpleasant odors, such as a filthy public restroom, or vomit, or manure. Right? Now, you've experienced extremely unpleasant odors. Do you want to permanently lose your sense of smell?
I definitely like having the color associations. It's an interesting thing to talk about, and it's kind of neat to have. And actually, once I realized that I could actually use colors to help me remember number sequences and things like that, that was actually kind of useful.
Synesthesia is not a disease. It's not a disorder. It's not something that is in any way a symptom of some other major problem. It's not something that you need to get rid of. It's an alternate way of perceiving the world.
For years I've been trying to understand, I'm not a synesthete, I've been trying to understand what it's like to have synesthesia. And the closest sort of analogy, which I think works, is something like this. If I were to show you a photograph of Barack Obama, you would immediately think Barack Obama. Even though those words aren't written anywhere there, just seeing his face, you would immediately think those words. An important thing to understand about synesthesia is it's not the same as a hallucination. So when a synesthete sees the letter T and experiences some sort of yellow in his mind's eye, it's not the same as a hallucination. He knows that the T is in fact in black ink and that there's not yellow out there. So it's not the sort of thing that would cause a car accident, and it's not the type of thing that would count as a hallucination or something one would have to worry about. So the most popular theory to explain synesthesia is that in different areas of the brain, you actually have cross-wiring. You have more wiring in a synesthetic brain that causes these areas to talk to each other more. I don't favor that theory. Instead, I favor a second hypothesis, which is that the amount of wiring is the same in everybody's brain. Instead, what's different is there's a slight difference in the balance of excitation and inhibition, such that activity here will cause activity here, because there's not enough inhibition of that activity. The reason I favor the hypothesis that the wiring is the same in everybody is because non-synesthetes can experience synesthesia. So for example, when people take drugs like LSD or DMT, they'll experience synesthesia. Also, many non-synesthetes, when they're very fatigued, right when they're falling asleep, somebody might slam a door and they'll see colors. So this implies that everybody is capable of a synesthetic experience. Also, for synesthetes, the amount of their experience waxes and wanes. So for example, when they are drunk with alcohol, or they're very tired, or they're very emotional or stressed out, their synesthesia tends to be much richer and more intense. And in other cases, when they're on certain drugs like antidepressants, their synesthetic experience goes down. And this has the feeling of something that is involved in inhibition in the brain rather than wiring, because there's no reason to expect that your wiring would change based on emotion and fatigue and so on.
So it doesn't matter if my Tuesday is blue, green, or yellow. The fact that it's anything may or may not mean something to someone else, but if it does, it doesn't matter if his is orange or purple.
So it turns out what we learned from synesthesia is that some people perceive reality in a different way than the rest of us. And this is probably true in many different ways that we haven't even discovered yet. Synesthesia just happens to be a very good inroad into understanding how this works and what's different about some people's brains that causes them to have a different reality.
And I believe there's a longer version of that on YouTube if anyone's curious about synesthesia. I did look it up while they were talking because I was curious. Synesthesia is not listed as a disorder in the DSM, and I knew it wasn't in the DSM, but it's not in the ICD, which is the one that medical doctors use. And so essentially, it's just something that happens. For most people who have it, it's not in any way disruptive. They can still function perfectly normally, and I like that it's not overly pathologized for that reason. The next one is just a short video from a 60 Minutes episode, kind of talking a little bit more about the background of synesthesia.
This is a story about ordinary people who see and feel things the rest of us don't. They have a rare brain condition called synesthesia, in which some of the senses, usually quite distinct, involuntarily fuse together, creating almost literally a sixth sense. Music is not only heard, it's seen and felt. Words can have flavors, and flavors can have color. It sounds bizarre, but it's more common than many people realize. And with the help of sophisticated brain imaging techniques, scientists are coming closer to understanding some of its hidden clues, and with that, perhaps opening a small window into the mysteries of the human mind.
They're not on drugs, they don't have any brain disease, they haven't had a stroke. They're born this way, and they're different from the rest of us.
Neurologist Richard Cytowic explored this surreal world in his book, The Man Who Tasted Shapes. He's documented hundreds of cases of synesthesia.
What is synesthesia?
You know the word anesthesia, which means no sensation. Synesthesia means joint sensation. And some people are born with two or more of their senses hooked together, so that my voice, for example, is not just something that they hear, but it's also something that they might see or taste or feel.
But how do we know that they're not making this up?
Well, first of all, it's been known to medicine and psychology for 300 years. This is an experience that happens to people. They don't do anything about it. The doorbell rings, they see, blue round spots.
And it's as real to them as
seeing the sky is to us. It's just that their reality is different than what we perceive.
Researchers like Richard Cytowic aren't looking for a cure. He thinks studying synesthetes offers remarkable clues to the mysteries of the human mind.
These people experience the world in a different way. Their senses are wired together differently. And so synesthesia, instead of being a quirk, an oddity that you just dismiss, it's a little peephole. And you move the peephole around and look through it, and you see this big expanse on how the mind works and how the brain is organized.
So I just like that description of it. It makes it a little bit easier to wrap your mind around. Another sort of interesting sensorial perceptive experience you can have is blind sight. So when you have blind sight, this is when you are aware that something's in your visual field somewhere in your brain, but you don't react to it. And if someone asked you, you know, is something there, you might say no. And they might have a feeling that something's there. They might be able to perceive the location of something, even though they say, what are you talking about? Nothing is there. You can ask them to guess and point to something, and they'll be accurate, but they will insist they do not see it. So it's a very interesting experience. And this can be caused by infarcts or tumors or damage in the visual cortex, or some of those stroke types we talked about briefly. So an ischemia or reduced blood flow, or an angioma, or which is a collection of abnormal blood vessels that results in abnormal blood flow. And so this next video again is short, but will give you a little bit of insight into the idea of blind sight.
You're now, I would claim you're now awake, you're wide awake, and you can be aware of my face, of the fact what sort of, that I wear a colorful shirt, that I talk with this Schwarzenegger-like accent. And so this I would call an operationalized definition of consciousness.
Christophe, why have you and Francis Crick, who I guess is best known for discovering the structure of
Just gonna turn it down a little swoosh.
DNA, chosen visual awareness to study consciousness?
We believe that because consciousness is an evolved property, that probably nature was discovered once during evolution, and the different forms of consciousness, i.e. visual consciousness, auditory consciousness, maybe even high-level self-consciousness, are probably just different variants of the same theme, the same mechanism.
So they've designed experiments to test visual consciousness. Eileen Wexler has normal vision, but this test simulates a phenomenon called unconscious vision, or blind sight, that can occur when there's damage to the brain's visual cortex.
So the world to them looks like this. They're blind in the left side of the visual field. And they go to the doctor, and the doctor says, well, can you see this? No. Well, can you guess whether it moves to the left or to the right? And the patient says, well, I'm blind. And the doctor says, well, guess. And the patient guesses left, right, up, down. And the patient is as surprised as the doctor, at least they were at first. And the point is that these patients, and there's now a quite substantial number of them throughout the world, they do not have conscious access to information in their blind hemisphere. Yet, if forced to respond, it turns out that something in their brain has information, because they can correctly guess, for example, the direction of motion. They can correctly, roughly point at a bright light. They can roughly say something about what colors is out there. No, it's very crude.
Senior research fellow Jochen Braun designed these blind sight experiments.
So let me show you the image one eye will see first. Okay, that's what it looks like. You have a background of squiggles all aligned.
Yes.
And there's this little area where the squiggles,
they're different.
They point at a right angle. Yeah. So I'll be asking you, where is this area, this different area?
It sounds easy, but during the experiment, the only thing I'm conscious of is a uniform pattern. Even though my left eye sees this, and my right eye sees this, my brain superimposes them. The theory is that some part of my brain will still know how to find the target, even though I'm not aware of it.
Now, the reason we do this is that the primary visual cortex, the first level of the brain which receives information from the eyes, receives information from left and right eye separately. So at this level, the brain can still figure out where the figure is.
Because it hasn't been superimposed.
It hasn't been superimposed, but at the next level in the brain, it's superimposed. So at that level, the brain is blind to where this figure is.
The glasses make sure that each of my eyes will see independently. Okay, here we go. I feel a bit foolish. I can't see any variation in the pattern. It seems impossible to find the target.
When we designed this experiment, we didn't know what would happen. And then the student who did the experiment came back and said, this is very strange. I'm rather good at finding the square, but I don't see it. So then it dawned upon us that this information, which does make it to the brain, at least at the first level, you can still use it, but you're not aware of it.
Their results haven't been consistent, but Aaron Rosen is one of the students who can sometimes locate the target 70% of the time, even though he's not conscious of it.
The theory, which has been advanced by others, not only by us, is that there are actually two visual systems, one ventral and one dorsal visual system with different functions. So the function of the ventral visual system would be to recognize things, to read, to see faces, to acquire visual memories, and so forth. And that visual system, the theory goes, contributes to your conscious experience. The other visual system, the dorsal one, has a different function, namely to guide your movements, your eye movements, and also your hand movements, or just locomotion if you're walking around.
Or say reaching for a cup of coffee.
Exactly.
Or a glass of beer. Here's another way of looking at the two kinds of vision, conscious and unconscious. No matter how you view this glass of beer, you know it's a glass of beer. But to pick it up and move it around, a separate visual system kicks in.
In some sense, we have within us a kind of robot that allows us to interact with the world that is controlled to some degree by our conscious visual experience. So our conscious visual experience marks this as a goal object, and then we bring to bear a robot that allows us to reach out and pick this up. Different computational demands.
So in a sense, that's unconscious vision.
It's unconscious vision, and it can, in some real sense, never be conscious. It's quite inaccessible to conscious report.
So, yeah, we can see stuff we don't even realize we can see, which is sort of interesting to try to wrap your head around. It's just this whole class, wrapping our heads around wrapping our heads, right? I'm looking for, they just made me think of, let me find it, is the various visual pathways. And so, yeah, they figured out that we don't just have one way of having vision, right? So there are multiple ways of having vision. There are multiple pathways. So you have this genicostriate pathway, probably butchering the Latin, apologies. And this is the part we looked at before where things are crossing at the optochiasm. And so you got everything for the right side and everything for the left side going together. And so again, going here, crossing over so that your primary visual cortex can integrate that information, make it make sense, basically. And then the next pathway relays through the lateral geniculate nucleus, or the LGN. And it sort of has this map of the visual field and allows you to sort of map things onto that visual field, and that helps you perceive where they are relative to each other, things along those lines. And again, this idea that we might have like an upper and a lower visual field, as well as a right and left. And then you have the pulvinar probably pathway, where the optic nerve leaves the eye and projects to the superior colliculus. And this is in the temporal and parietal lobe. So again, vision is not just in the occipital lobe. It detects stimuli and helps us orient to the stimuli. So this may be more of that sort of like unconscious vision that they're talking about. And here you can see those two pathways taking the same information from the eye, but using it in very different ways. The other thing that I'm not finding my slides for, so I'll just talk through it. Let me scroll up one more time and see if I can find them. chat through it. Okay, is there are different types of vision or visual systems. And the occipital lobe is part of all of those. I'm just making sure this was actually a dry erase. So one of those is vision for action. And this is the visual processing that we need to direct movements, particularly like I need to move my arm there, I need to move my whole body back over to the screen, right? That allows us to do so. And I will just put this on something neutral so it's not confusing. This allows us to do things like reach, duck, catch things. And it is in the parietal visual areas in that dorsal stream. So again, not in the occipital lobe, in the parietal lobe. Then you have action for vision. And this is where you're searching for like a part of a target object and attending selectively to it. This is visual scanning. This focuses in on eye movements, but also uses a lot of the parts of our brain that focus on selective attention. We use action for vision in particular when we are looking at faces. So we focus in on people's eyes and their mouths. Then there's visual recognition. And just like that sounds, that's recognizing objects. And this is in the temporal lobes. So again, not all in the occipital lobe. And visual space. This is in both the parietal and the temporal lobes. This has to do with spatial location. So both location of an object relative to a person. So if I'm looking at that trash can across the room relative to myself, and that is called egocentric. Ego here not meaning I think so much of myself, but meaning about myself, essentially. And then it can also be a location of an object relative to another object. So for example, because I have visual space, this concept of visual space, I can look at this projector thing here and I can tell it has two different little heads and a base, even though they kind of cross over each other, because I have that ability. And that is called allocentric space. The relation of different objects to each other. And then there's visual attention. What are we going to pay attention to? And I'm just going to abbreviate it. In our vision, because we see a lot of stuff all the time, right, that we're looking at. So like if I just stand here right now, I can see the lights, the speakers, the fans. I can see outside a little bit out of my peripheral, right? I can see the clock. I can see behind y'all to the little lounge area out there, waiting area. That's a lot to take in. I can only see all 4, 5, 6, 7 of you here today, right? Your lovely smiling faces, or trying not to fall asleep faces, no judgment. Right? And so that's a lot for me to take in all at once. And so visual attention helps me to focus. It helps me to say, okay, right now I need to focus on the screen or my notes to make sure I'm giving them this information correctly. Right now I need to look around and maybe make some eye contact with people, trying to engage them, right? And so it also allows us to not be overwhelmed by all that visual information all at once, right? Last thing I'll mention, eye-wise, is a scotoma. And this is something that can actually go wrong with the occipital lobe. And so you have small blind spots in your visual field, but they're not because of cataracts or floaters or anything on your actual eye. It's because of small lesions in your occipital lobe back here. The nice thing is you're often unaware because your brain, when it can, will spontaneously fill in this information with information from the rest of the occipital lobe. And the brain is used to doing that from the blind spot that we mentioned previously. Alrighty. Touching briefly again on some of the other senses. So there are various somatosensory receptors for the bodily senses. And again, we tend to, when we think of the bodily senses, just think about touch, right? But in fact, we have a lot going on in the bodily senses. So pain and temperature is nociception. It's unpleasant stimuli. So we have free nerve endings for pain, which is like our sharp pain, our dull pain. It's a slow response. And as I mentioned, it's like tissue damage, and in particular, the dendrites that stick off the axon body of the neuron. And then we have free nerve endings for temperature as well. Heat, cold, and again, slow, and again, damage, sunburn, frostbite, things along those lines. Hapsis is fine touch and pressure. And so you can see there's various versions here. Touch, flutter, and vibration are pretty rapid, and the stimulus is just pressure against whatever part of our skin. We can also have them for steady skin indentation, that's a slower one, or our hair receptors, right? I mean, people talk about getting goosebumps or goose flesh, right? Where your hair kind of stands on end and you get little bumps, right? That gives us information. Sometimes that something's dangerous. Sometimes that's something just like really emotional is going on for us. And then proprioception, or that bodily awareness. We've got muscle spindles. Oops, sorry, back of here, where the muscles stretch. We've got Golgi, tendon organs. We can tell the tendons to stretch. And we have joint receptors, the movements of our joints. These are all very rapid, and it's movement stretching the receptors. And so this movement might be caused by us, by our brain telling our muscles and our joints and our tendons to, but it could also be caused by something we don't. Like you fall down the stairs and your leg twists the way it's not supposed to, right? Or someone runs into you. You're taking steroids and one of your muscles rips or your tendons rips because it can't keep up with your muscles. All that kind of fun stuff. Alrighty. And then this gets more at what happens when that stuff gets up to the brain. So we have this somatosensory cortex right here. And so you have the dorsal root ganglion neurons that are responding to that fine touch and pressure, to the joint, tendon, and muscle change, and to pain and temperature. Those are going to relay to the spinal cord initially. And then that ventral spinothalamic tract receives input from pain and temperature neurons and comes together in this pathway. The dorsal column nuclei relay those. So again, it's not one big neuron going up there. There are various other nuclei relaying the information. And then that primary somatosensory cortex receives that information. The thalamus relays sensory information to that somatosensory cortex. Oh, sorry, I jumped over here. So the primary somatosensory cortex is last. You would next get axons that carry it to the thalamus, then the thalamus relays it over to the somatosensory cortex. Sorry, they didn't really put these in any discernible order from when you're talking. And so your brain, that somatosensory cortex, maps onto various parts of your body. You may have seen the homunculus before. It's sort of terrifying, right? Where it's basically representing how much of that part of your brain is dedicated to various parts of your body. So we have a lot on our hands. This isn't representing as much, but we also have a lot on our feet. We don't think about it as much, but we need our feet to be paying attention. We trip on a crack or we step on a rock, right? Our feet have to be able to react pretty quickly. And then there's a lot on the mouth and the tongue because speech is so important to our communication as humans. And some of the other parts, not as much. So that's the homunculus, if you've ever seen that before. It's lovely and can also be nightmare inducing. So, you know, balance is the last sort of somatosensory submodalities for these bodily senses. As I mentioned, we've got those semicircular canals in the inner ear. There's also something called the otolith organs that respond to acceleration, and they pay attention to head positionality. Now, the last thing I will touch on, and then I'm actually gonna let y'all out a little early today, just because I talked fast, is perception. And in intro books, and in this book in particular, perception often gets the short end of the stick, right? So there are like 2 slides about perception on here versus 58 about sensation. And perception is basically your subjective experience of the stimuli after they're into your brain. And it's influenced by context, but why do you all think we spend so much time and pages focusing on sensation and then so little about perception? Again, there's no one answer. Yeah. Yeah, I think that makes a lot of sense, right? So if our differences are so individualized, right, that it's really hard to interpret. Any other thoughts? Anyone on Zoom have anything you want to unmute or stick in the comments about this? Yeah, I think it's that. Oh, go ahead.
You're a little far away when you said the question. Can you repeat it?
Yeah, no problem. Why do we spend so much time talking about sensation and then perception gets like 2 slides?
Perception is incredibly varied.
Exactly. Yeah, it's varied. We also don't understand perception as much. So I think that's a piece of it, honestly. And some people think that because it's subjective, it shouldn't be as important. But like, how much of human experience is subjective, right? That sort of becomes the question. Again, it's something interesting to sort of think about going forward. But certainly, like I said, I got through the sensory system really quickly. So we will actually wrap up early today. Quiz 1 covers everything up through today, I believe. So make sure that you take that online, or if you want a hard copy, I have them. And ask me if you have any questions. If you're coming to class in person or going to be attending on Zoom, make sure you have your case study book accessible, because we'll do another case study worksheet on Wednesday. Thank you all, and hopefully no one's offended you're getting out early. I'm guessing no.